The next topic is the kidney, and a bit like the liver, we first of all need to look at its gross structure before looking at the functional units of the kidney, which are called nephrons, which you may remember from GCSE. So dealing with its gross structure, here's a nice diagram which shows aspects of the gross structure of the kidney. The kidney can be, if you do a cross section of the kidney, it can be divided into various regions. There's an outer layer here which is called the cortex, and then there's an area inside the cortex which we call the medulla. The kidney is supplied with blood from the aorta and the renal artery, and then blood that's been processed by the kidney then returns to the bloodstream in the renal vein. The filtrate that's produced by the kidney is funneled by a region called the pelvis of the kidney, which is here. It's funneled into a tube called the ureter, which takes the urine, as it's called now, to the bladder. As an aside from one of the last topics we did, sitting like a little woolen hat on top of each kidney is the adrenal gland. It also has an outer region which is called the adrenal cortex and a middle region which is called the adrenal medulla. The adrenal medulla being responsible for producing adrenaline, whereas the adrenal cortex produces corticosteroid hormones, which we studied in the hormones topic. Here's another diagram which shows much the same, except in this diagram it orientates where our functional units of the kidney are. Functional units of the kidney are called the nephron, which you may remember from your GCSE study. But just going through the cross section here, again we have the cortex, renal cortex as it's known, which runs out around the outside. Okay, and then we have the medulla region, which is in the middle around here, and as I say, the renal pelvis, which is where all the filtrate from all the nephrons drains into this renal pelvis area, which is then funneled into this structure here, which is the ureter, which takes the urine off to the bladder. Now the functional units of the kidney, analogous to our liver lobules that we looked at in the liver, in the kidney the functional unit is called a nephron. And there are millions and millions of these nephrons in each kidney, and they're arranged such that part of the nephron can be found in the cortex, okay, and then bits of the nephron stretch down into the medulla, and eventually the filtrate that's produced by the nephron empties through the medulla, this structure here called the collecting duct, into the renal pelvis, and then all the millions of nephrons empty their filtrate into this renal pelvis, which then funnels it into the ureter. Here is a nephron itself, okay, with some structures which you might remember from GCSE. I've got another diagram which will look at this, which perhaps makes it easier to understand, but we've got a knot of capillaries here, which is called a glomerulus, a structure called the Bowman's capsule, a squiggly tubule here, which is called the proximal convoluted tubule. Convoluted means squiggly or windy, proximal means close, and then we've got a long tube which stretches down into the medulla. You can see that on this diagram here, but on this diagram here, that's what it is there. It's called a loop of Henle, and for the purposes of this we talk about a descending loop, limb of the loop of Henle, and also an ascending limb of the loop of Henle. We'll look at that in a little bit more detail. And then we have a thing called the distal convoluted tubule, okay, which is also a winding tubule, which is distal, further away from the start of the nephron, and the distal convoluted tubule then connects with a collecting duct, and the collecting duct also passes through the medulla and empties any filtrate into the renal pelvis. So here's the collecting duct here in this diagram, running down and running into this region here, which is called the renal pelvis. So looking at the nephron in a little bit more detail here, here's what you need to know. You need to know keywords, word glomerulus here. You need to know this word glomerulus, which is a knot of capillaries. And the knot of capillaries is in association with a sort of blind end of a tube, which is called the Bowman's capsule, which I'll show you that in a minute. And then the Bowman's capsule leads into a thing called the proximal convoluted tubule, which leads into the loop of Henle, okay, which has got a descending limb and an ascending limb, okay, and then the loop of Henle leads into the distal convoluted tubule, which leads into collecting ducts, which empties into the pelvis, the pelvis funnels urine into the ureter. Looking at that diagrammatically, I'll just quickly go through that again. So here's a schematic diagram of what one of these nephrons looks like. So we have this knot of capillaries here, which is surrounded by this blind-ended tubule, which is called the Bowman's capsule. And that leads into the proximal convoluted tubule here, which then leads into the descending limb of the loop of Henle here, which then leads into the ascending limb of the loop of Henle, which then leads into the distal convoluted tubule, which then leads into the collecting duct here. Looking at some of these areas in a little bit more detail, we'll move on to the next slide, which so describe and explain the production of urine with reference to the processes of ultrafiltration and selective reabsorption. Okay, so there's quite a lot of detail we need to know here. This is a rather elegant diagram here of what's going on in the Bowman's capsule. So first of all, we have a network of capillaries, okay, which are covered in these funny purple cells, which I'll talk about in a minute. But the network of capillaries, or glomerulus, okay, or glomerular capillary, which is tend to call it the glomerulus, is fed by an arteriole. So the arteriole supplies blood to this network of capillaries, okay, or glomerular capillaries, and the whole thing is called a glomerulus. You'll notice that once the blood has passed through the glomerulus, it then joins with another capillary, okay, and we call that well, not a capillary, sorry, wrong word, it's an arterial, and we call that the efferent arterial. Okay, so it's rather odd in that we have an arterial feeding a capillary, which feeds another arterial, because this usually feeds a venule. But in this case it's an arterial, and we call these, the incoming one we call the afferent arterial, and the outgoing one is called the efferent arterial. Now, one of the key things to notice here, if you notice the width of this afferent or incoming arterial, it is wider than the width of the efferent arterial. What this means is that blood being pumped under ventricular pressure or systolic pressure through here, as it goes into, there's more blood coming in here than there is blood coming out of here. What happens is that that creates a higher pressure inside the glomerular capillaries than would normally be the case in a capillary bed. So there's high pressure in this capillary. Okay, and that is one of the key influences in the whole process of forming urine, is that we have, it's a filtration process, and I'll come to the filtration process in a minute, but this word ultra basically means under pressure, and the pressure is created due to the blood pressure pumping blood into the capillaries and the fact that there is a differential between the width of the incoming arterial, that's wider than that. Okay, so pressure develops ultra in this capillary space here. Okay, so that's the ultra bit of the word ultrafiltration explained. Now, the complicated bit to visualise, or this diagram does actually represent it rather well, is the idea of the filtration. The Bowman's capsule is constructed that it's got an outer layer of cells, here, which are normal flattened squamous epithelial cells, which form a watertight structure around the outside of the Bowman's capsule here. The inner lining of cells are quite interesting, in that the inner lining of cells are continuous with the capillaries. Okay, and here's one of these cells here, and these cells have got a special name. They're called podocytes. Podocytes, okay, as outlined there. And the podocytes wrap themselves around the capillary. And I always think of the analogy of a podocyte being a little bit like a hand wrapped around, let's say, a tennis racket. And essentially the key thing is that there are gaps between the fingers, okay, these finger-like projections, which are actually called pedicels, okay, and it's the gaps between these things which are actually the important bit, because that allows easy movement of molecules from the bloodstream into the Bowman's capsule, which surrounds the capillaries. Because if it was a continuous layer of cells, if it was squamous epithelial cells wrapped around these capillaries, it would be very difficult for any of the filtrate from the blood to actually pass into the nephron. So the gaps created by these podocyte cells allows easier filtration. If we were to look at that in a little bit more detail here, we have blood coming along a capillary through here, and there are three things to consider, three layers which separates the blood in the capillary from the filtrate in the Bowman's capsule. The first thing to consider are the endothelial cells of the capillary itself. Okay, And like the capillaries we studied in f214 with respect to tissue fluid formation, these endothelial cells have got pores in them, they've got holes in them. Okay, sometimes the word fenestrated is used. They have little windows in them. Okay, and these little windows enable molecules to pass out of the capillary. Remember, they're not big enough for big cells, blood cells, to be able to pass out of there. Okay, but they are large enough for molecules such as water and oxygen and so on to pass through these fenestrations or pores. Wrapped around the endothelial cells of the capillary, there is a structure called a basement membrane. Now, this basement membrane is a confusing word because one thinks of membranes as phospholipid bilayers in biology, but this membrane isn't a phospholipid bilayer. It's made of a meshwork of collagen fibres. And these collagen fibres act as a filtration device. So they don't allow large molecules or cells to pass through, but they do allow smaller molecules such as water, salts, urea, oxygen and so on. They do allow those molecules to pass through, but they don't allow the larger molecules to pass through. And then surrounding the basement membrane we have these cells which form the lining of the Bowman's capsule around the capillaries, which are these special cells called podocytes, okay, which I made by this analogy about a hand with fingers gripping a tennis racket. So they wrap round the capillary, and it's actually the gaps between the fingers, if you like, or they're actually the fingers are called pedicels, but it's the gaps between these which are the key thing. Okay, they form what we call filtration slits, which allows molecules that have passed through the basement membrane to easily pass into the Bowman's capsule. So, just summarising all of that on the notes that you have. Okay, so you've got to understand what the term ultrafiltration means. The word ultra is to do with pressure, and that's caused by the incoming arterial having a wider bore than the outgoing arterial, as I've just shown you on the diagram. Filtration basically means that the small molecules, water, salts, glucose, amino acids, urea, etc., can leave the capillary in the glomerulus and pass into Bowman's capsule. Okay, and there are three things to consider that these filtered substances that I've just outlined here have to pass through three layers, if you like, between being in the blood and actually getting through into the filtrate in the capillary. The first thing are the pores in the capillary endothelial. So the capillary is made of cells called endothelial cells. You should remember that from f214. And these have pores in them. Okay, that word fenestration I used refers to the pores in these endothelial cells of the capillary. Wrapped around the endothelial cells is this structure called a basement membrane, which is like a meshwork of collagen fibres, which acts as the filtering membrane, which prevents large molecules and cells, etc., passing from the blood into the filtrate. Then wrapped around the basement membrane are these specialised podocyte cells. Okay, and the gaps between podocyte cells create what we call filtration slits, which allow filtered substances to easily pass into the Bowman's capsule. The next bit to look at is what we call selective reabsorption. This is quite complicated. This occurs in the proximal convoluted tubule, which is the first of the winding tubules that passes from the Bowman's capsule. If I look at this diagram here, if you can imagine the proximal convoluted tubule, which we saw on the last slide, has the filtrate passing down here, okay, and the proximal convoluted tubule is made of a succession of these types of cells here on either side. So there'll be cells here, cells over here, and the filtrate runs down here. What happens is, as the filtrate passes down, substances that we don't want to lose in our urine, such as glucose molecules, amino acid molecules, etc., water molecules even, are reabsorbed back into the bloodstream. So they pass through the cells that line the proximal convoluted tubule, and they pass through the back end of that cell into the bloodstream. Okay, so a lot of the molecules that we've just lost from the bloodstream are very quickly reabsorbed. But the cunning thing about all of this is that it's selective, is that we only reabsorb the molecules we want, and we don't reabsorb the molecules we don't want. So if we look at this cell, there are various features that we need to consider with this cell. First of all, you notice that we've got these structures here which increase the surface area of the cell in contact with the filtrate. These are called microvilli, which we've come across before. So their job is to increase surface area because we've got an exchange surface here, and therefore we want a large surface area for that to exchange things with. These cells, I'll have to draw these in because they don't appear in this diagram, but these cells would also have lots of mitochondria in the cells because they're heavily involved in active transport, which we'll see in a second. So what happens is that molecules that we want to absorb are absorbed by a special type of transport which you didn't study at f214, and I'll quickly explain it. It's called sodium cotransport. And what happens is that the molecules, be they glucose or amino acids or whatever they are, we've got glucose here as an example, are absorbed into the cell along with a sodium ion, okay, quite a heavily charged sodium ion. This helps change the conformation of this carrier molecule, which helps pull the glucose molecule through the membrane. So the glucose and the sodium pass through together by a process called sodium cotransport through one of these cotransport molecules here. So the glucose gets into the cell, as does the sodium. And what we do is the sodium is pumped by active transport out of the cell here. The idea being that there's always a steep concentration gradient of sodium in the filtrate and sodium in the cell, which means the rate of diffusion through here is going to be greater because of the higher concentration gradient. The glucose passes by facilitated transport through a glucose carrier molecule out of the other end of the cell too. Other features of the cell, you'll notice that the cells are connected to each other. So the cells here and here are connected to each other. Okay, those are called tight junctions between cells, and that's to prevent any leakage of water or molecules between the blood and the filtrate, or the filtrate and the blood. So it produces, if you like, a fairly waterproof layer of cells, so that molecules have to pass through these special carrier molecules. And there will be different carrier molecules for different things. So there would be each amino acid, for example, would have its own specific carrier molecule. Each of the 20 different amino acids would have specific co-transport proteins which would take those amino acids into the cell along with sodium ions. So that's how that works. It's quite complicated. Okay, so I'll just quickly go through that again. So the microvilli is using this as a prompt. The microvilli increase surface area. Okay, the membranes have these co-transport proteins which take molecules such as glucose or amino acids into the cell along with sodium ions. Sodium ions are then pumped out actively to lower the intracellular sodium concentration, which means there's a steep concentration gradient between the filtrate and the cell, which means that there's a higher rate of diffusion of sodium into the cell along with the glucose. Glucose passes out of what's called the basolateral membrane, which is the back end of the cell, which is associated with the blood capillary here. So the glucose passes out of that into the blood capillary. So what we've done is reabsorb glucose from the filtrate back into the blood here. I've also pointed out that these cells have lots and lots of mitochondria because they need to produce lots and lots of ATP to be able to pump these sodium ions into the blood. So the next thing we have to look at is explain, using water potential terminology, the control of water content of the blood with reference to the roles of the kidney, osmoreceptors in the hypothalamus, and the posterior pituitary gland. Okay, so we just looked at in the previous slide of how a number of substances such as glucose, amino acids, are selectively reabsorbed from in the proximal convoluted tubule. In actual fact, lots of water and salts are reabsorbed in the proximal convoluted tubule, but there is now a special mechanism which enables us to absorb more water and more salts from the kidney filtrate. So, things to consider here is that in the descending limb of the loop of Henle, water passes from the tubule to the medulla cells. These are the cells surrounding the loop of Henle. Okay, and that happens purely by osmosis, simply because the water potential in the filtrate is greater than the water potential in the cells, or the tissue fluid that surrounds the cells of the medulla. So that's what's happening in the descending limb on the way down. And on the ascending limb, what we do is we pump sodium and chloride ions out of the filtrate, okay, which actually reduces the water potential in the medulla, which draws water out in the descending limb. I'll show you that in a second. So if I look at this diagram here, just to summarise what I've just been saying, so this is the descending limb, and as we can see, as the filtrate passes down the descending limb, water moves out of the filtrate. The reason it moves out of the filtrate is because the water potential is higher in the filtrate than it is in the surrounding cells. So as the water moves down here, as you can see, it becomes progressively more concentrated. So 300, whatever that 300 is, to 600 to 1000, okay, in terms of concentration of sodium chloride. So we have a high concentration of sodium chloride at the bottom here, and as we turn the corner, what we start doing is we start moving sodium chloride ions out of here. We pump sodium chloride ions out. Okay, so the cells here have cells with lots of mitochondria producing ATP, which pumps sodium ions out of here, which reduces the concentration of sodium ions as we move up here. Okay, but the movement of the sodium chloride ions into this medulla region makes it more concentrated, higher salt concentration here, which reduces the water potential, which allows the water to move out of here. Okay, so it's quite a complicated process. So that's what happens in the descending limb and the ascending limbs of the loop of Henle. Descending limbs, water is removed. Ascending limb, sodium ions are actively pumped out. So the other thing, and the fact that the descending limb and the ascending limb are so close together creates what we call a hairpin countercurrent multiplier. And what that essentially means is that it's the active removal of the sodium ions in the descending limb actually creates a water potential gradient for water to move out of the descending limb. Okay, and that requires the descending limb and the ascending limb to be close together. In adaptations of kidneys of different mammals have different lengths of loops of Henle. Ones that are animals that live in very arid conditions, desert rats and so on, they tend to have very long loops of Henle, whereas mammals that live in an environment where there's lots of water, such as a beaver, they have very short loops of Henle. So, moving on. Once the filtrate has moved out of the loop of Henle, what then happens is it passes through the distal convoluted tubule, and you don't really have to know much about the distal convoluted tubule at all. In fact, you don't have to know anything at all about it, so we won't mention it. But we do have to know what goes on in the collecting duct. Okay, the collecting duct provides us an opportunity to regulate the amount of water that we absorb from our filtrate. So the filtrate passes from the ascending limb up through the distal convoluted tubule, and then it passes through the collecting duct, which actually isn't a diagram of here, but it passes through the collecting duct, and then that eventually goes into the renal pelvis, which takes the urine into the ureter. The collecting duct is lined with cells which can alter their permeability to water. So if your water levels are low, the cells become more permeable, which allows more water to be reabsorbed, so we conserve water. If your water levels in your blood are high, what happens is that we make our cells of the collecting duct impermeable to water, so all the filtrate passes out into the renal pelvis and into the ureter, and we would make copious amounts of very dilute urine. Whereas when our water levels are low, we absorb more of the water, so we produce small amounts of very concentrated urine. The way it works is this. Just rearrange all of these for a second. If you can imagine that this structure here is a cell lining the collecting duct. When water concentrations are low, you produce from your pituitary gland a hormone called ADH. Okay, which will just draw us a little blob there. And ADH passes through the blood and binds to an ADH receptor cell. So this is cell signalling again. A receptor cell on the cell surface of the collecting duct cell. Inside the cell are vesicles which contain protein channels which allow water to pass through membranes. And these channels are called aquaporins. Okay, an aquaporin, so it's a water channel. So here are the water channels here and here. Now when they are in this vesicle, the cell membranes are not permeable to water. When ADH binds to this ADH receptor, what happens is a second messenger is produced in the cell, our old friend cyclic AMP. So there'll be adenyl cyclase here, which will convert ATP to cyclic AMP, a bit like we saw with adrenaline in the past. And what the cyclic AMP does is it causes the vesicle to move to the cell surface membrane. I can't actually do that in this little animation, but what does happen is that as the vesicle moves, these aquaporins are then inserted into the cell surface membranes of the cell. And what that means is that water can now pass through into the cytoplasm and out through the other side. So ADH, which was represented by this red blob here, causes the water channels to be transferred from inside the cell into the cell surface membranes, which makes this cell permeable to water, which means that we would imagine this was the filtrate here. So when that happens, ADH binds to the molecule and binds to the receptor. The aquaporins are inserted in the cell surface membranes, so water will start to move along a water potential gradient, okay, from the filtrate into the medulla, because remember the collecting duct passes through the medulla, and also because the water potential in the medulla is low because of all that sodium chloride in there, water will pass from the filtrate into the bloodstream, which will reduce the amount of water you lose in your kidney filtrate, i.e. you'll produce less amounts of urine and more concentrated urine. When the ADH isn't around, it isn't being produced, so ADH doesn't form there, the cyclic AMP isn't produced, what happens is that these water channels will then be corralled back into the vesicles in the cell cytoplasm, and the cell membrane will now be impermeable. Okay, so water will not be able to pass through here. So that's what happens when you've got lots and lots of water in your blood, is that no ADH is produced, therefore these cells here are waterproof, and therefore any water that gets this far in the nephron will just pass through in the filtrate, and go into the kidney. Go into the kidney pelvis or the renal pelvis. So you'll produce copious amounts of very dilute urine. The other thing you've got to be aware of here is the presence of how ADH is released. Okay, so we talked about what ADH does. ADH is is produced actually by cells in the hypothalamus, okay, which is in the brain, and it's produced in special cells called neurosecretory cells, okay, whose cell body is in the hypothalamus, and they have an axon which stretches from the hypothalamus down to part of the pituitary gland called the posterior pituitary gland. Okay, and I'll just find a picture of that. Here we go. So if you can imagine this as a neurosecretory cell, here's the cell body of the neurosecretory cell, which is in the hypothalamus, and here's the axon of this neurosecretory cell, which stretches from the hypothalamus down to this structure, which is just above the roof of the mouth, called the posterior pituitary gland. So ADH, which Americans call vasopressin, is produced in the cell body, protein synthesis in action, so lots of ribosomes, endoplasmic reticulum, etc. It's then bundled into vesicles, which are then transported, okay, down the axon, which stretches all the way from the hypothalamus down to the pituitary gland, and then they're released by the pituitary gland. Okay, and there'll be special receptor cells called osmoreceptors, which will detect levels of water in the blood as it passes through the hypothalamus. So what will happen is that if water levels go down, that will send a message to here, which will stimulate ADH production. ADH gets transported along the axon into the posterior pituitary gland, where it's released into the blood, and then it circulates in the blood, where it binds to ADH receptors on collecting duct cells, which increases the water permeability of those collecting duct cells by inserting aquaporin molecules, which we talked about before, aquaporin molecules into the surface of the membranes.